Hello and welcome again to Child Psychology. In the previous topic, you have been introduced to various perspectives in understanding child psychology. Some of the experts believe that socio-cultural aspects plays an important role in child development. For example, Vygotsky and Erikson. While others, such as ethologists, focus on biological factors which determine behavior. So, which one is which? Does biological aspect, or in other words, heredity or genetic loading, or environmental aspect such as socio-cultural influences determine the development of children? Let us discuss about it. Meanwhile, this is your learning objective. You must know how new lives are formed, understand building blocks of human being, what is hereditary mechanism, and explain about environmental factors. Lastly, you must know of how both of these factors interact with each other. The basic building blocks of human being are cell, chromosomes, and DNA. Let us examine one by one. We are human beings made of about 37 trillion of cells. There are about 200 types of cells. One important cell that you must know is sex cell, the sperm and ovum. Other cells are called somatic cells. For example, look at your hand. It has somatic cells. Look at your hair. It has somatic cells too. Basically, inside the cell, there are several organelles such as mitochondria, cytoplasm, and the most important for heredity is chromosomes. Somatic cells are diploid. Each has 46 chromosomes or 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes. But our sex cell is haploid, which consists only 22 autosomes and 1 sex chromosome. When they pair up, they become a full chromosome. Our chromosome is made up of nucleic acids. We have DNA and RNA. DNA, or deoxyribonucleic acid, is a double-stranded and made up of backbone of sugar, that is deoxyribose and phosphate, and the nucleobase. For DNA, the nucleobases are adenine, or we give a symbol A, guanine, we give a symbol G, cytosine for C, and thymine T. Now, what about RNA? RNA is ribonucleic acid, which is single-stranded, with sugar of ribose and nucleobase of A, G, C, and U. And U is for uracil. What you must understand is that the sequence of nucleobase is very important because it determines the genetic code. Different sets of genetic code are translated into different proteins. These proteins are basic elements of your hair, nails, enzymes, hormones, muscles, cartilage, skin, blood, and many more. So, genetic codes are very important to determine who you are. Let us watch how our DNA and its genetic code functions to produce protein.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
What is conception? Let us recall. Let's say this is a mother and this is a father. The mother has 46 XX and the father has 46 XY. Mother's sex cell, that is ovum, contains haploid 22X chromosomes and sperm contains 22X or 22Y chromosomes from the father. Conception occurs when the sperm meets the ovum and becomes a single cell called zygote. A baby boy has 44 XY and a baby girl has 44 XX chromosomes. Remember, father's chromosome contains DNA with his genetic code, and mother's chromosome contains DNA with her genetic code. Each genetic code serves as a template for hereditary information called genotype. Let us look at this diagram. C1 is a genotype of curly hair and it is a partial dominant. Only those with double C1 will have curly hair. A mother who has straight hair carries only one C1 and C2. Her ovum carries either C1 or C2. The same goes for father. He has straight hair because he carries genotype C1 and C2. But his sperm carries C1 or C2. When sperm that carries C1 meets ovum that carries C1, their offspring will have both C1. Hence, he or she has curly hair. Other siblings will only have straight hair if they have only one C1. The observed properties of genetic makeup, such as curly hair, are called phenotype. Can genetic expression change with the changes in environment? The answer is yes. Let me explain. If the dough is the genes and the artists are environment, exposure of similar genes to different environment can produce different end results. Different behavior may develop with similar genes. This process is called epigenesis. Feral child is an extreme example of how environment could change the genetic of human. Feral child is a human child who has lived isolated from human contact from a very young age and has little or no experience of human care or behavior. They have marked changes in cognition and behavior following the isolation. Some of them adapted to animals' behavior if they lived with animals. Before we finish, let us hear opinions from other experts on heredity or nature versus environment or nurture in relation to child development. Listen and learn how they argue or which one is more important.
We are all born with certain physical characteristics determined by our genes. But does our DNA dictate how we act?
Genes may very well determine how we behave in given situations.
Researchers say they are learning now that genes help control how we interpret our environment.
There has to be some kind of innate circuitry in place that allows us to create culture and acquire culture and do the learning.
But can environment influence which genes are triggered?
What really matters is which genes are turned on and which genes are turned off. And different genes are turned on and off in different circumstances.
That leads to debate over the role parents play in influencing their children.
Parents aren't the only aspect of the environment. There's also the surrounding culture.
Some believe the stronger social influences happen outside the home.
Children live in separate worlds, and each world is very important, but there's very little carryover from one world to the other. And what is carried over is whatever is genetic.
Controversial author Judith Rich says how well a child turns out is up to the child, not the parent. Others find that troubling.
It would encourage parents to be either passive or hopeless or helpless in the face of certain challenges that they would assume were genetic.
As the debate rages, what's clear is that as scientists study DNA, they are learning more about ways to fight disease and perhaps understand better what makes us tick. I'm Dr. Dina Dunn.
Okay, learners, now we have finished discussion about this topic. See you in next topics. Thank you.